In this video, we will discuss the main features of prokaryotic cells. The first cells were prokaryotes, and they were found as actually fossils. Here on this image, you can see what's called microfossils, the remnants of the original prokaryotes from which life started, the last universal common ancestor. Shape-wise, they indeed look like modern prokaryotes. The oldest prokaryotes are about 3.5 billion years. That was the time when the life actually started. In the modern days, we actually have those ancient relics in the form of stromatolites. Stromatolites are mats of cyanobacteria that can be found in different areas around the world, mostly in the warm climate, for instance, in the Shark Bay near the coast of Australia. These cyanobacterial mats, layers basically of cyanobacteria, can trap mineral deposits, building these amazing structures. These are actually layers. These structures are built by layers of bacteria. There are stromatolites right now that originated 2.7 billion years ago. So there are organisms, well, not organisms, but structures that pretty much are as old as almost as old as life on Earth. So how can we date? How can we know when a certain structure started? How old is it? For this, we can use carbon dating. Living things incorporate carbon-12 more than other isotopes, which explains why they have greater levels of carbon-12, compared to non-living things. One can perform the analysis of relative concentrations of the isotope carbon-12 in fossils, and this analysis demonstrated that the first organisms that could fix carbon, the first organisms that could convert inorganic carbon to organic one, existed as long as 3.8 billion years ago. So in order to identify different organisms and characterize the history of their development, scientists use biomarkers. Biomarkers are various organic molecules that aren't formed independently but are produced by living cells. It is not easy to find specific biomarker, but it is known that some hydrocarbons derived from the tails of fatty acids found in the membranes were identified in the ancient rocks, and these findings demonstrated that first bacteria, cyanobacteria, are at least 2.7 billion years old. As a matter of fact, the most recent findings of biomarkers in solid rocks demonstrated that the origin of life can be traced to as far back as more than 3.5 billion years. So prokaryotes are the oldest group of organisms on Earth. They are the most abundant in the world, and they are fairly simple in terms of their structure. Prokaryotes were abundant way before eukaryotes arrived, roughly 1 billion years before the first eukaryotes can be detected. Right now, the entire prokaryotic diversity is greatly understudied. We know less than 10% of prokaryotic species. Prokaryotes fall into two domains: bacteria and archaea. So this is an example of the microscopic view of bacterial cells, and these are bacterial cells growing on the agar plate. So there are multiple lines of evidence suggesting that um, first prokaryotes were pretty old. Two main lines of evidence are microfossils and stromatolites, and also presence of biomarkers in the fossils. So what is the difference between bacteria and archaea? Well, any cell, including prokaryotes, contains plasma membrane. So bacterial plasma membranes contain lipids that are not branched, and these lipids use ester bonds to connect to phosphate groups. Membranes in archaea, on the other hand, formed on the backbone of glycerol, but linkages are ether, not ester. In addition to that, hydrocarbons in archaea are branched, unlike in bacteria. The advantage of having four ether bonds rather than ester bonds is that extremophilic microorganisms, organisms that exist at very high temperatures, can withstand those high temperatures. Some bacteria have peptidoglycan cell wall. Archaeal cell walls can contain peptidoglycan or can be formed by other carbohydrates. Both bacteria and archaea have the single origin of replication. However, the mechanism of replication of archaeal DNA is much closer to the mechanism of replication in eukaryotes. Same can be told about the process of gene expression in archaea, which is more resembling of the gene expression in eukaryotes rather than in bacteria. These are, as we mentioned before, are the most abundant organisms on Earth. Less than 10% of prokaryotes, as we mentioned as well, have been described only. They can live in absolutely diverse habitats, from frozen lakes in Antarctic to extremely hot geysers in Yellowstone National Park. But they all share the same defining features. They are unicellular, they have small cell size, they have single circular chromosome, they reproduce asexually and facilitate the increase in genetic diversity by the use of horizontal gene transfer. In addition to that, prokaryotes lack internal compartmentalization. Prokaryotes can use a variety of metabolic strategies to receive energy and carbon. All these strategies can be grouped into four main categories. Photoautotrophs, they use light as the source of energy and can convert inorganic carbon into organic compounds. Chemoautotrophs, which use chemicals as the source of energy but still can convert inorganic carbon into organic compounds. Photoheterotrophs, which use light as the source of energy and have to use inorganic carbon as the carbon source. They can't fix carbon. And finally, chemoheterotrophs. Most bacterial chemoheterotrophs, they use chemicals as the energy source and utilize organic carbon as the carbon source. They have extremely fast reproductive rates. They are essentially the fastest dividers in the world, but they use binary fission rather than mitosis. Due to the rapid cell division and quick accumulation of mutations, they show enormous genetic diversity and very quick evolutionary rates. So early classification of prokaryotes was based on the staining and the phenotypical characteristics such as whether they are photosynthetic or non-photosynthetic, whether they can move or cannot move, whether they form colonies, whether they form filaments, or they exist as strictly individual cells, whether they form endospores or can divide by transverse binary fission. And finally, whether this particular microorganism that is about to be classified is important as a human pathogen. Right now, the classification rests on the molecular characteristics of prokaryotic microorganisms, amino acid sequences of proteins, the percentage of GC pairs in the genome, which is unique for each species, similarities between the nucleic acid sequences tested by nucleic acid hybridization. So closely related species will have more base pairing between nucleic acids. And finally, sequencing of genes that are present in the genome and RNA sequencing as well. All of this allowed scientists to group prokaryotes into two very distinct domains, bacteria and archaea. This classification was based on the sequencing of ribosomal RNA genes, particularly 16S ribosomal RNA, and later whole genome sequencing finalized this definition of new largest taxa in the classification of living organisms: eukarya, archaea, and bacteria. You can also see that these three domains of life are evolutionary, they have very interesting relationships. Archaea, which is a prokaryote, is evolutionary closer to eukarya rather than they both to bacterial cells. Let's talk about the shape of these organisms. Let's start with bacilli. Bacilli are rod-shaped microorganisms. Many of them are gram-positive. Some of them are gram-negative. Gram-positive bacilli, such as bacillus, can produce a unique structure known as endospore, which we will talk about later. Some of them can cause human disease. Actinobacteria, shown here, is common in soil, but also in the human mouth can be found some of the species. They form filaments and actively produce antibiotics that we can use in the clinical practice. Spirochetes is another shape here. It's a spiral shape of bacteria. Some spirochetes can be human pathogens. For example, Treponema pallidum, the causative agent of syphilis. So we already mentioned that they can be bacilli. They also can be cocci, circular shaped cells. Streptococci form chains of circular shaped cells, while staphylococci form clusters. Many bacteria have cell wall, which gives bacteria shape and protects cells physically and mechanically. Cell wall in bacteria prevents the cells from exploding in the hypotonic environment in which bacteria typically live. Finally, based on the structure of the cell wall, bacteria can be categorized as gram-positive or gram-negative. Due to the different structure of the cell wall, gram-positive and gram-negative bacteria have different sensitivity to antibiotics. Some bacteria also have a structure known as S layer. So let's compare gram-positive and gram-negative bacterial cell walls. Gram-positive cell wall is relatively thick and contains teichoic and lipoteichoic acid. Gram-negative cell wall, on the other hand, is fairly thin, and on the outside of the cell wall is an outer membrane. Inside of the outer membrane, you can find structures such as lipopolysaccharides and porins. Lipopolysaccharides, lipoteichoic acid, and teichoic acid molecules can all serve as the determinants of the disease in humans. So, to reinforce, gram-positive bacteria surrounded by the membrane and the thick cell wall made out of peptidoglycan polymer that consists of carbohydrates and amino acids, while gram-negative bacteria is surrounded by the cell wall, thin peptidoglycan layer, and the outer membrane containing lipopolysaccharides and porins. Now, there's a technique called gram staining that allows us to determine which bacterial species we're dealing with. First, bacteria are stained with the primary dye called crystal violet, which stains all cells purple. Then bacteria are treated with iodine, which is a mordant, which facilitates the formation of the complex between the crystal violet and the iodine, which sticks very well to the cell wall of gram-positive microorganisms, but not gram-negative ones. After that, cells are treated with alcohol, which removes crystal violet iodine complex from gram-negative bacteria, making them colorless, but not from gram-positive ones. And finally, safranin, which is the secondary stain, is added to the sample. It has no effect on already purple gram-positive cells, but stains gram-negative cells pink. And this is what you can expect to see under the microscope if you have a mixture of gram-positive purple and pink gram-negative cells. Another type of cell wall, which is sometimes found in archaea, is S layer. It's paracrystalline layer, which is formed for the most part by the proteins and to some extent carbohydrates, which exists outside of peptidoglycan or outer membrane. For the most part, its function is adhesion. Another interesting structure that is found in some bacterial species is capsule. We mentioned capsule before when we were talking about the Friedrich Griffith experiments. So capsule is the layer of usually carbohydrates found in some bacterial cells. Capsule is a protective structure. It may include proteins, but it protects bacteria from immune system and also helps in the process of attachment. Some bacterial cells have flagella. This is the attachment responsible for the motility, for the movement. This is the helical structure that is embedded into the membranes and cell wall. Here you can see this. And it actually is rotated very much like the axle of the propeller is rotated within the motor. Flagella consist of the protein named flagellin, and, as you can imagine, is involved in the movement. Another external appendage that can be found in some bacteria, pili. Pili are rather short compared to bacteria structures that aid bacteria in either attachment, or they can also help bacteria to exchange genetic information. Few bacterial species, not many, but quite a few bacterial species, can also form a structure known as endospore. Endospore is a protective, again, structure in which bacteria develop a wall around their genome in order to protect it from the harsh environment. So basically, lack of nutrients, desiccation, presence of toxic chemicals can stimulate endospore formation. When conditions surrounding bacteria get better, endospores can germinate into a fully functional vegetative cell. Bacteria that cause anthrax, tetanus, botulism, gas gangrene, ulcerative colitis can all form endospores. In some bacteria, you can find the folds of the inside of the cell. It's not the membrane-bound organelle, it's just the folds of the plasma membrane. The function of that structure is, for the most part, to increase surface area, so different cellular processes can be more active, let's put it this way. Very interesting type of bacteria are cyanobacteria, which are photosynthetic. They can be found on the roots of the plants and in the oceans. They largely responsible for what we call algal blooms, though they're not algae. And they also are very active in the fixation of nitrogen. Some gamma proteobacteria can also be photosynthetic, and there are some pathogens among that group. Now, what about the cell structure of bacteria in terms of, we figured that they may have capsule, they can have flagella, they can have pili, they can have endospores, but they don't have internal membranes as the membrane-bound organelles. All they may have are just folds of the membranes. Finally, bacteria have a region called nucleoid where DNA is located. And of course, all bacterial cells have ribosomes. You can notice that antibiotics act on bacteria because bacterial ribosomes, antibiotics that target bacterial ribosomes, that inhibit the action of bacterial ribosomes, target exclusively bacteria but not fungi. The answer to the question shown on the slide is because fungi are eukaryotes, so antibiotics that target bacteria would be ineffective against fungi. So we mentioned that nucleoid is where bacterial DNA can be found. And bacterial DNA forms single circular chromosome. In addition to that, bacterial cell can also contain small circular DNA molecules known as plasmids. Bacterial ribosomes are smaller than the ones in eukaryotes, and they are different structurally in terms of the protein and RNA content in them. This is why we, as a society, developed some antibiotics that target specifically bacterial but not eukaryotic ribosomes. So prokaryotes lack sexual reproduction. To ensure the genetic diversity, prokaryotes have three types of horizontal gene transfer: conjugation, transduction, and transformation. All these three processes are also observed in archaeal cells. In conjugation, plasmids that encode some advantageous information. Remember, these plasmids are not required for the cell to survive. These plasmids are transferred through so-called sex pili. Okay, so for instance, in E. coli, F plasmid, so-called fertility factor, if plasmid is present in, let's say, this cell, the cell is called F plus cell. If plasmid is absent, it's F minus cell. So the plasmid is transferred from F plus to F minus cell. It is not a form of bacterial reproduction. It's the form of the gene exchange. So here you can see the transfer of the F plus of the F plasmid from F plus cell to F minus cell. The result of this product is that both cells, the old donor and the recipient, they both become F plus. Another source of genetic diversity in bacteria, of course, is mutation, but it plays a lesser role than conjugation, transduction, and transformation. So F plasmid in some cases can integrate into bacterial chromosome, which resembles the process of crossing over in eukaryotes. The mechanism behind it is homologous recombination. Cells that can carry out such incorporation are called high frequency of recombination cells, and every time F plasmid is integrated into chromosome and then excised out of the chromosome, the inaccuracies in the excision may result in the plasmid picking up some genes from the chromosome, and now we call it F prime plasmid, which is different from the original F. So here's an example of the recombination. So F plus cell here. Here you have the recombination site. So plasmid gets incorporated into the chromosome, and then it gets excised, but you can see that in some cases, if excision isn't precise, the plasmid can pick up some other parts of the chromosome. Transduction is the process of the gene transfer carried out by bacteriophages, viruses of bacteria and archaea. Generalized transduction is carried out by the phages that perform what's called a lytic cycle. Practically any gene can be transferred during generalized transduction. Basically what happens, those viruses, bacteriophages, they package bacterial DNA and then infect another bacterial cell and transfer that bacterial DNA that they have packaged. On the other hand, specialized transduction is characteristic for the phages that carry out lysogenic cycle. In this case, phage can incorporate its genome in bacterial chromosome. The excision, imprecise excision of bacterial genome of so-called prophage, results in the addition of bacterial genes to the prophage DNA. Only specific host genes can be transferred via specialized transduction. So here's an example of the phage that infects bacteria. Its new phages are synthesized, and you can see that this particular one packaged the fragment of bacterial genome. Infection of a different cell by that phage results in the generalized transduction of the genome from that cell into this one. And the donor DNA is incorporated into the chromosome of the recipient cell. Transformation can be natural or artificial. Natural transformation can occur in many bacterial species, such as Streptococcus. We talked about it when we talked about Friedrich Griffith experiment. In the process of transformation, DNA that was released from the dead bacterial cell is picked up by another live bacterial cell and then incorporated into bacterial chromosome. Um, it is absolutely normal process. This cell dies, and the fragment of the DNA from the dead cell is picked up by another cell and gets incorporated into the chromosome. In some cases, we actively use artificial transformation. Some species don't normally undergo transformation, so scientists can transform those bacteria in the lab. It's very widely used to clone genes and express proteins artificially by transforming E. coli. E. coli, or prokaryotes, I'm sorry, are extremely abundant in soil, in water, on the human skin, guts, animals. Most of the prokaryotes are beneficial or neutral, and without the original prokaryotes, life on Earth would be impossible. Why? Because they are the key, um, they have two key functions, decomposition and photosynthesis. Decomposition provides organic compounds, and photosynthesis originally generated oxygen on Earth. Many prokaryotes live in symbiosis. Symbiosis is pretty much living together. So in case of symbiotic relationships, they can be mutualistic when both parties benefit, for example, nitrogen-fixing bacteria that deliver nitrogen to the plants, or bacteria that produce an enzyme called cellulase, allowing animals to digest cellulose. And then commensalism and parasitism. In commensalism, only one organism benefits, another one does not receive any benefits, but it isn't being harmed either. In parasitism, on the other hand, one organism receives the benefits and another one is, generally speaking, harmed. Prokaryotes are widely used for a production of drugs, so-called biopharmaceuticals, bioremediation, for instance, of the mines, insect control, that doesn't rely on synthetic chemicals and therefore much less dangerous to humans, and to some extent they were contemplated to be used in biological warfare, but these projects were abandoned due to the inefficiency. Of course, they can cause human disease. So, what is a pathogen? A pathogen is a microorganism that can cause a disease in humans. In the period before antibiotics, a lot of bacterial infections that are now absolutely mild were lethal. Now, in the antibiotics era, many bacterial diseases reemerge due to the development of antibiotic resistance in bacteria. Pathogens can be spread by air, by respiratory route or aerosols, by contaminated water, in other words, by fecal-oral route, or by vectors such as ticks and mosquitoes. In the early 20th century, infectious diseases of different sort killed roughly 20% of children before age of 5, so it was a major factor in mortality. Improved sanitation and the use of antibiotics improved the situation considerably, but in recent years, as we mentioned before, many bacterial diseases appeared back. This is the short list. Please do not memorize the species for each disease, but you heard about them: anthrax, botulism, chlamydia, cholera, gonorrhea, Hansen's disease or leprosy, pneumonia of different etiology, typhoid fever due to the salmonella plague. They all still are detected in the modern days. We're going to mention some of those diseases, and we're going to start with tuberculosis. It's the respiratory infection caused by Mycobacterium tuberculosis. Up to a third or a half of human population have been exposed to tuberculosis at least once in their life. Tuberculosis results in about 9 million new cases every year, and it is the highest risk for developing tuberculosis are for people who live in crowded conditions. It is multi-drug resistant in some cases, total drug resistant, and it causes pretty much a very severe pneumonia. Lyme disease. Another human pathogen known as Lyme disease is caused by bacteria known as Borrelia burgdorferi, shown here on this slide. It is transmitted by ticks, and its first symptom is so-called bullseye rash at the site of the bite, results in a fever, and may, if untreated, may lead to encephalitis, meningitis, arthritis, and other complications. You can see that Lyme disease is more common in the U.S. Northeast, surprisingly not in Ohio. The main transmitters of the reservoirs for Lyme disease are mice and deer. So the different and the transmission rates can be associated with the changes of available food for those animals. Dental caries is another example of bacterial human disease. So plaque that is formed on the teeth, it consists of biofilms, thin layers of bacteria. Streptococcus sobrinus, a microorganism that lives in these plaques, ferments sugar, producing acid, which degenerates the enamel of the teeth. Example of bacterial infection that affects stomach is peptic ulcer that is caused by Helicobacter pylori. How do we know that peptic ulcer is caused by a bacteria? It can be treated with antibiotics. Bacteria are also an important cause of sexually transmitted diseases. For example, gonorrhea. You can see that in the United States, the rates of gonorrhea are on the decline, as well as the rates of syphilis, is caused by a microorganism called Neisseria gonorrhea. It is still one of the very prevalent diseases in North America, but you can see that chlamydia sort of takes over. Gonorrhea is transmitted via the exchange of vaginal and penile secretions and can even pass from a mother to child when the child passes through the birth canal. Chlamydia is another example of STD. It is second most common sexually transmitted disease in the US after the papillomavirus. Incidence has skyrocketed because often chlamydia infection is fairly asymptomatic, so it is hard to detect until it starts causing serious troubles. Finally, syphilis, caused by Treponema pallidum. It is transmitted only sexually or vertically from mother to child, and it has 4 distinct stages. So primary syphilis is characterized by highly infectious and painless chancre on the reproductive mucosa. Secondary syphilis is characterized by mucocutaneous rash that manifests itself both on the skin and mucosal membranes. Tertiary syphilis is when it becomes latent. The patient is not infectious anymore, but at this point, bacteria start attacking internal organs. And finally, quaternary syphilis, which is sometimes merged with tertiary, is the stage at which the damage caused by the bacteria, damage to internal organs, damage to the connective tissue is visible.